Welcome back to Chapter 9, Part 2. Let's learn a little bit more about mitosis, how it evolved, how it's regulated, and exactly what cancer is. So starting out here, if we're going to talk about how mitosis evolved in the first place, we really have to take a closer look at how our closest non-eukaryotic relatives divide. So let's take a look at the process called binary fission that happens in bacteria cells. Prokaryotes like bacteria and archaea, they're going to do binary fission. Some eukaryotic cells do a version of this, but we'll talk about that in a second. So let's look at the details here. Remember that in bacteria and archaea, you only have one chromosome. It's a single chromosome. And it's going to start to make copies of itself at this region called the origin of replication, which you can see on the diagram right here, the origin of replication. And so the origin of replication is actually attached to the plasma membrane. And that's very different from what happens in chromosomes in eukaryotic cells because, our chromosomes are in the nucleus when they're going through replication during interphase. That's just not the way it happens in prokaryotes. So this single chromosome, it starts making copies of itself. And as it's making copies of itself, the origin starts to move farther and farther and farther away from each other itself. So there will be two origins. So here's the original origin, and it's starting to make a copy of itself right there. You can kind of see that in green if you've got good color vision. And if you have a good computer, you can kind of see that in green. Those origins start to separate from each other as replication is occurring. Now again, we still haven't learned how replication occurs yet, but just kind of imagine that DNA is opening up and making copies of itself, making sister chromatids. The cell is elongating, it's becoming longer and longer and longer, which is what's causing the origins to pull farther and farther and farther away. Eventually, the DNA is completely copied and the origins are pretty far apart from each other. There's an origin there and there's an origin right about there. And at that point, the cell can pinch in or it can make a cell plate depending on what kind of bacterial cell we're talking about. In any case, the plasma membrane will be pinching in here and the new cell wall is going to be deposited in order to make two completely new cells. The process is much simpler than what happened in mitosis. And one of the reasons it's so much simpler in binary fission is because in binary fission, you only got the one chromosome. In mitosis, in humans, you got 46 chromosomes you got to keep track of. And so things are going to have to be much more complicated to deal with all those chromosomes. So I told you that we'd talk a bit about the evolution, and we do know a few things here. Prokaryotes evolved before eukaryotes evolved. So mitosis probably evolved from the process of binary fission. We do know that in some protists, like here's some diatoms, these are single-celled eukaryotes. These are dinoflagellates. They're really cool. Both of these are marine organisms. Some yeasts also do it this way, where the cell division looks like something kind of in the middle between binary fission and mitosis. So, like, if you look here at how the dinoflagellates do it, their microtubules are attached to the plasma membrane, the nuclear envelope doesn't disappear, and there's no centrosome there. It's just kind of all just attached to the plasma membrane. Similar thing happens in diatoms and some yeasts. These groups also, the nuclear envelope does not disappear, it stays intact, and the thing happens here is that the kinetochore, the microtubules, they actually form inside the nucleus. So dinoflagellates and diatoms kind of do it a little bit differently, but it's really not mitosis. It's some interesting middle of the road of evolution process that happens in those organisms. So the next concept that we're going to hit is talking about how the cell cycle is regulated. It's not some free-for-all where whatever cell wants to divide goes and divides. That's just not the way it works. The frequency of cell division varies greatly among the different cells in your body. For example, your blood is going to fully replace itself about every 4 months. Your skin is replacing itself almost every single month. You've got new skin, completely new skin. Your brain is going to take about a year, your lungs 2 to 3 weeks, things along those lines. So there's variation in how frequently different cell types go through mitosis. This is really important because these differences end up having to have very, very specific kinds of regulation at the molecular level. And so we'll talk about that too. Finally, we're going to talk about cancer cells and how cancer cells manage to escape the control system of the cell cycle. And so we tend to say that cancer is the cell cycle out of control, or you could say it's mitosis out of control. So let's learn a little bit about what control actually means. And control is associated with this concept of the checkpoint. Checkpoints are a series of events that have to happen that control the cell cycle system. It's like, if you think about it, it's kind of like, one of those, an older washing machine where you have to turn the dial and it sets things and so forth. This analogy doesn't work as well if you have a nice digital dishwasher, but that's okay. Maybe you've seen these before. So the cell cycle control system is regulated in a couple of different ways. It's got internal controls and it's got external controls. And if you think about, again, this is interphase all the way through here, G1, S, and G2, and then here's mitosis. We have 3 basic checkpoints. We have the G1 checkpoint. And all that a checkpoint means is like, you're like, okay, am I ready to move forward? Can I do the next thing? Yes. Okay, check. Good, we can move on. We have the G2 checkpoint that happens right at the end of G2 and the beginning of mitosis. By the way, the G1 checkpoint happens in the middle of G1. There is no checkpoint here during S, just so you know. But if things didn't go right in the DNA synthesis phase, then it'll get caught here at the G2 checkpoint. And then there's the M checkpoint, and the M checkpoint happens in the middle of mitosis. I'm going to go into this a little bit more detail for you, but basically what this cell is saying is, did everything go right? If everything went right, okay, let's move on. If things didn't go right, then we stop. So let's take a look at the G1 checkpoint. The G1 checkpoint is probably the most important checkpoint. Sometimes it's called the restriction point if you're talking about mammal cells, because at this point, a cell will either be allowed to continue through this interphase, move into the S phase, or it will not receive the go-ahead signal. A big old stop sign will be put up there, a stoplight, and if it doesn't get the signal, then what we say is that it enters a period of stasis where the cell just stays in that G1 phase. But because it's staying in there for a really, really, really long time, we give it the name G sub 0 or G0 phase. Sometimes you might hear it called the G naught, N-A-U-G-H-T, naught. But yeah, that's like English, British English. So we're going to stick with G0 phase. If it does complete the G1, if it gets the green light, then it's going to go ahead and make copies of DNA in the DNA synthesis phase. So that's what we mean by the checkpoint. An internal signal occurs at the M phase checkpoint. what happens here is that the checkpoint is looking to see whether or not anaphase can begin. And anaphase can't begin unless the microtubules are attached to the kinetochores, and of course also you need some non-kinetochore microtubules. So it's saying, did we connect up here properly? Did we get all of our microtubules associated with kinetochores? And if so, then we're going to continue through the M checkpoint and say, yes, okay, we're good. We can move on to anaphase. If we don't pass the M checkpoint, if things don't look quite right, then that cell will die. And that's a good thing, because you want your chromosomes to separate properly. Checkpoints in the cell control system are regulated by something called growth factors. Now, you might look at this word growth factor and think, oh, this has to do with growing. And okay, sure, right? Growth is one of those things that's can only happen if mitosis is happening properly. So growth factor is not a bad term, but once you're fully grown, you still do have growth factors regulating your systems, your checkpoints. So what happens is you might get these external signals called growth factors binding to receptors in your plasma membrane. And that plasma membrane sends a signal. I know we've talked about this before. If you're having trouble remembering this, you might want to go back and review that chapter on parts of the cell. We have this signal transduction pathway, and it's sending the signal down through here. Eventually that signal is going to enter the nucleus and say yes to the G1 checkpoint, we're in the clear, or no to the G1 checkpoint, nope, you're going to enter G sub 0. But again, this is all regulated by growth factors. Another example of an external signal is something called density-dependent inhibition. That is quite a mouthful. Let's break that down for just a second. Density, that's how much stuff is in a small space, right? Density-dependent inhibition. So what this means is that if cells are crowded in high density, then the cells are going to be inhibited from dividing. They're not going to be allowed to divide. So you can see what that might look like. Here's a dish filled with cells, and when they get to a certain place where they have plenty of cells and all the cells are touching each other, then the cells would say, oh no, we're dense enough, we got enough cells here, we don't want to go any further. So you would enter G0. Most animal cells also exhibit something called anchorage dependence, and anchorage is more than just a city in Alaska. It is it's when things are attached to something. So anchorage dependence means that if they have to be attached to something, either inside of your tissues or inside here, this dish, in the lab, you might have this happening in a dish, in order to divide. So we're not going to divide unless we're anchored. That's what that means, which is kind of interesting. Remember I told you about how red blood cells are formed in bone marrow. Red blood cells don't form themselves inside the bloodstream. They form in the bone marrow where they have some anchorage. Cancer cells, interestingly, exhibit neither density-dependent inhibition nor anchorage dependence. So let's take a closer look at what that means. Oh, before I move on to what cancer means, I just want to notice that a cell here, a little patch of cells had been removed, and if they were scraped away, then all the ones nearby will start dividing, and that way the gap fills in. That's like, how your body repairs itself if you've been cut, for example, something along those lines. But I did promise to talk to you a little bit about cancer, and so we're going to spend several slides talking about cancer. Normal mammalian cells, they have this density-dependent inhibition, right? And they're going to form a single layer, and this picture is a single layer of cells. and it might grow, it might fill in those gaps depending on how many nutrients are around, or if you got some growth factors, or if it's got a substrate for attachment. But cancer cells, they're like, they don't care. Cancer cells don't care. They will continue to divide beyond a single layer. You can see how this depth of field here, this is flat, has a much deeper field because they are overlapping on each other. They're not exhibiting anchorage dependence. You can see this one's kind of floating here. And they don't have density-dependent inhibition. So that's a bad thing, because that means that cancer cells are not being regulated in the same way that other cells are being regulated. So they don't respond to the normal signals that regulate cell cycle. As I said before, they're not going to stop just because they don't have growth factors. They actually make their own growth factors. They're not going to stop growing just because it's too dense. They don't care about density. They're not going to stop growing just because they don't have anchorage. They don't need anchors. So that's what is like the kind of basis, the background of what makes cancer grow and grow and grow. And of course, I'll show you in a couple of slides here what that means for the body. But for now, let's take a look at just a basic growth. And tumor is a growth. So when we say cancer, we often think of this word tumor, and they're not always the same thing. So let's see if we can get some differentiation here. In a lab, and we say cells in culture, when we say in culture, we're talking about like in a petri dish in the lab. They can divide, if they can divide indefinitely, if they can divide forever and ever and ever, we say that those cells have undergone a process called transformation. They've transformed from normal behavior into like the cancer werewolf behavior. It's full moon, they've transformed, right? They're not the same cells. But there is something really cool that happens in your body, and that is that your body's immune system actually sees and eliminates a lot of tumors that start to form. So tumors are just these masses of abnormal cells dividing and dividing and dividing. And they're surrounded by normal cells, but they just form this big kind of lump here, or tumor here, because they're dividing so much. Now, you can have abnormal cells that form a tumor or a lump, but that are not considered cancer. So we call those kinds of lumps benign tumors. Older dogs sometimes get these big fatty lumps on their bodies. Those are benign tumors. Women sometimes get cysts in their breasts. Those are benign. They're not cancerous. It's only cancer if it's malignant, if the tumor is malignant. And to be malignant means that it has to invade surrounding tissues. It also has to undergo metastasis, and metastasis means the cancer moves from one area of the body to another area of the body. So again, in order to be cancer, it's got to be malignant. At the very least, it has to be malignant, meaning invading surrounding tissues. So a benign tumor would have a capsule around it, and these cells in here might be abnormal, but if they're surrounded by normal cells and they don't invade those normal cells, then it's a benign tumor. It's a malignant tumor if you no longer have that connective tissue capsule and the abnormal cells start to kind of intermingle or move into the region where the normal cells are. Again, an extreme form of this is metastasis, where it's not just invading surrounding tissues, the nearby tissues, it's going into all other parts of the body. You might have heard that somebody might have breast cancer, but it's that breast cancer has moved to their liver, for example. It's still breast cancer, but it has metastasized to the liver, and that is not a good sign. That's full-blown cancer. So another way to look at this is to actually look at some breast tissue here. And what we have is showing you is a tumor. And the tumor, it was just one single cell that got a mutation in it. And that started this, ignoring the cell cycle information. And so you get this tumor. But if it's just a lump right there, if it's staying right there, and if it's surrounded by that capsule that we just talked about, then it's benign. But if it starts to invade other tissue of the breast, then that is cancer, and that's a malignant tumor. Now, the malignancy can then pass into either the lymphatic system or into the blood vessel system, and it can go throughout the rest of your body. So that would be metastasis. And while most cancers, well, I guess I shouldn't say it that way. I should say a small percentage of cancer cells can metastasize to far distant regions of the body, but most cancer cells do tend to stay local, and therefore can be operated on with surgery or something along those lines and can be dealt with that way. And that's, if that's where you are, you're lucky, just having to have some surgery. But there are other ways to treat cancer. You've probably heard of them. They can be treated with high energy radiation, and high energy radiation damages DNA. If it's done well, and these days it can be very precise, it can pinpoint exactly where that beam is going to go, and it can just kill those cells that have the cancer cells, the mutant cells. That's great. But if you've got, or if you suspect that it has metastasized, then you're going to use chemotherapy. And every chemotherapy drug that I'm aware of is a drug that interferes with a specific step of the cell cycle. So I grabbed this diagram off the internet. I just thought it was a really good diagram. It's oriented a little differently. So here's interphase G1, there's S, there's G2, and there's mitosis. And so, and they put the different signals in a slightly different place than your book does, but that's okay. One of the things that, if you've ever met anybody who has cancer or know somebody who has cancer, they have probably had one of these types of chemotherapy drugs. And one that's very frequently used in breast cancer is tamoxifen. And tamoxifen acts as a hormonal interrupter that prevents the cell from leaving G1, and therefore, if the cell is trying to grow but it's not growing, if it's a cancer cell, then it's going to not be able to continue growth, and those cells actually do divide. Remember that cancer cells are dividing and dividing and dividing just all the time. So if you can stop that division, then you can stop the cancer, and then you can kill those cells. So that's good. But of course, one of the side effects of chemotherapy is that it's going to kill all cells that divide frequently. We've talked about several of them already. We talked about how your skin cells divide fairly frequently, as well as your red blood cells. So people who go through chemotherapy often get anemic, meaning they don't get enough oxygen moving around their body because some of those red blood cells have died off and they haven't been replaced because the chemotherapy is killing off those dividing cells in the bone marrow. Also, hair loss is very common for cancer patients who are undergoing chemotherapy because it's killing off the skin cells. Remember, skin cells are some of the ones that divide very frequently, killing off the skin cells, and skin cells some of those are specialized to make hair. And so if the skin cells that were dividing stop making your hair follicles, then your hair will keep growing a little bit, but then it won't have anything to anchor onto, and it'll die. It'll fall out, basically. So that's what's happening with cancer. Loss of control of the cell cycle, just kind of wrapping this whole thing up. We do know that the cell cycle and cell cycle signaling have given us a lot of help in understanding how to treat cancer, and that medical treatments for cancer are becoming more personalized depending on what mutation you've got. Is this a signal in this cancer that's ignoring the M checkpoint? Is it a signal that's ignoring the G2 checkpoint? Is it a cancer that's ignoring the G1 checkpoint? So we can use personalized medicine to help treat with the very specific mutations that have happened for the very specific types of cancer that you or a loved one might have. So I hope that you have gotten some good information here about mitosis. I know that we often spend a lot of time talking about the process of mitosis, but mitosis is such an important concept, and that I hope you have gotten some good information out of this that you might be able to use in your everyday life. Thanks for listening, and I'm going to move on to talking about Chapter 10, Meiosis.